शुरू कर दें। Okay. Good morning, everyone. So today topic is immunological basis of pathobiology and therapeutics in PBC. So PBC is a chronic autoimmune liver disease where the immune system destroys bile ducts, causing bile buildup and liver damage. So I am going to talk about how PBC is with the immune mediated injury. And immune mediated injury, how it destroys the Beck's biliary epithelial cells and induce the intrahepatic bile acid accumulation. Is there any molecular mimicry in this system? And is there any importance of the nuclear receptors? And finally, the therapeutic options. So basically, hepatocytes are and the cholangiocytes are constant attack with the PAMPs, DAMPs, xenobiotics, and through the portal venous blood flow. And when the hepatocytes are surrounded by these exogenous and endogenous mediators, they replace these mediators, they activate the few signaling pathways, and then it changes these mediators, DAMPs and PAMPs, and present to the cholangiocytes. Cholangiocytes, when they accept these exogenous and endogenous DAMPs and PAMPs, they start reacting these DAMPs and PAMPs. And this reaction of cholangiocyte cause the pro-inflammatory microenvironment. However, this pro-inflammatory microenvironment is not solely done by the cholangiocyte. It's an interplay of the hepatocyte, fibroblast, cholangiocytes, even leukocytes, and they start the repair response. Most of the time, there is a resolution of this injury in 95% of the patients. However, in the genetic susceptible patients, there is a loss of tolerance and persistent repair goes on, and this leads to progressive biliary injury and inflammation, and then end up into the cholangiopathy, cholestasis, ductopenia, fibrosis, and the neoplasia. Actually, cholangiocytes really are the immune tolerant. How they do it? Whenever they see the DAMPs and the PAMPs through the TLR signaling, they start secreting the cytokines, and there is a homeostasis. How? Because TLR through the mid 88, there is a production of one kinase, interleukin 1 receptor associated kinase M, which balance the cytokine production, which is here IRAC-M. If IRAC-M is not there, then there is a too much of cytokine production, and then there is a loss of immune tolerance. Otherwise, cholangiocytes maintain the homeostasis. Other than the TLR signaling, PPAR ligands and receptors, they also start recruiting the immune cells for the to avoid the injury. So there is a balance of CD4 and CD8 T cells which they recruit, and sometimes they have even the immune tolerant CD4 and CD8 organs. Ultimately, cholangiocytes express the TNF alpha and MCP1 and CXCL16 for the recruitment of the more CD4, CD8 cells. So they maintain the immune tolerance homeostasis. But there is a breach of homeostasis. Why? This portal area is all overexpressed with the CXCR3, which is the migratory molecule that it leads to the T cell trafficking and differentiation of even the CXCL9, 10, which are the chemokine ligands, which attracts the leukocytes. And when too much of the leukocytes are there, which is rich in the portal tracts, then there is an imbalance of the breach of tolerance. Now they have observed that CD8 effector, not the CD4, CD8 effector memory phenotype are resistant to the apoptosis, and they start accumulating in the portal tract near the cholangiocytes, and then these CD8 T cells start making the injury. In latest Davis et al. 2024, there is a Nature Communication paper where they have shown very beautiful example of the cell-in-cell structure process in the liver, where they have showed that there is an invasion, in the sense big CD8 T cell is entering to the cholangiocyte, and those CD8 T cells are positive to the E-cadherin, CD103 positive. So basically E-cadherin positive. So what happened, the biliary epithelial cells have the E-cadherin junctions, and these activated CD8 T cells express E-cadherin. They start making the contact with the BACs, and then these cells make the contact and form a strong E-cadherin beta-catenin interaction within the BAC. You know that the beta-catenin is almost ubiquitously present in all membranes of the cells. So, but however, they have also showed, it is a big paper where they have done the confocals, they have done the cell cultures, and they have shown that how CD8 T cells change their cytoskeletal rearrangement and to make the invading and make the cell-in-cell structure, illustrating that E-cadherin on T cell facilitates this unique entry into the epithelial barrier. It means the BAC have the now CD8 T cell inside the BACs. So what happened? The T cell internalization with the BAC was more frequent for those expressing CD8 in PBC liver tissue than expressing CD4. This phenomena does not hold true with the CD4. It holds true with the CD8 T cells. And therefore, in 2025, there is a phase 1 clinical trial. There is a one molecule OP724, which is a CBP catenin antagonist, whose tolerability was well demonstrated in patients with advanced PBC. Now, other than the CD8 T cells, whenever the pro-inflammatory cytokines induce the dendritic cells, there is, of course, differentiation of the B cell, CD4, CD8, and the CD4 differentiate into the TFH. Latest in 2018, and then further in 2025, the Adam and other groups have said that in PBC, there is a generation of pathogenic TFH. What is the pathogenic TFH? Are the T follicular helper cells which make the contact with the B cell and make the antibodies against. So how they are different? So in hepatology communication in 2018, they showed that in the normal homeostasis, IL-2, which is secreted by the T cells, they differentiate the TFH cells with the PD-1, ICOS, and the TCR. But in the autoimmune diseases, including the PBC and PSC, there is a SNPs mutation in the IL-2 RA and the receptor, and then they start giving the pathogenic TFH, and even the T regulatory cells also in normal homeostasis, they regulate the TFH, they inhibit the overproduction of TFH. But when there is a mutation, these Treg cells do not stop and they start producing the pathogenic TFH in the PBC. So, in the 2018, they demonstrated that in PBC, in comparison to PSC, there is lot of CXCR5 positive TFH cells, and even CXCR3, which I already told you in the portal tract, there is a chemokine receptor which start attracting the more and more cells, and then there is a more in the PBC even than the PSC, and they have correlated that in the cirrhotic patients, when the cirrhotic patients are there, there is more in the PBC, in the TFH, pathogenic TFH. So there is a immune infiltration of CD8 T cells and the TFH together. So these TFH were even positively correlated with the AMAs, IgM, and not with the IgG. Now, another paper in 2025 scientific reports, they showed that endothelial cells, they start expressing the CPE and VWF factor involved in the immunopathogenesis. How? They did the single cell sequencing of the liver endothelial cells, total tissue, and then they found that this is the control and this is a PBC. This blue is the endothelial cells. You can see that in PBC, these endothelial cells were significantly reduced. Other than these, in PBC, there were the 7 kind of endothelial cells through the single sequencing they have shown. And they have shown that there is a CPE. This is a cytopathic element. And with the expression of VWF, endothelial cell types, that was more in the PBC patients. And these cells, endothelial cells, start interacting with the CD74 and MIF, which is more present in the liver tumor. And CD74 serve as a receptor for the pro-inflammatory cytokines. So directly or indirectly, these endothelial cells, which are very specific in the PBC, they start producing the pro-inflammatory cytokines, and even MIF, which is a counteract with the anti-inflammatory effect of glucocorticoids, which you use in the clinics for the PBC. So these hormones therapy do not work efficiently in PBC because of these special endothelial cells generation in the PBCs. There is another way of immune injuries to the back. Whenever there is a immune injury in the back, what happened? The mitochondrial inner membrane, which is the PDC-E2, which is a pyruvate dehydrogenase cell, E2 mitochondrial inner membrane, it comes out. When it comes out, it starts expressing on the surface. What happened when this PDC-E2 is coming out in the back? It starts expressing. It is now again inviting the other T cells that you come and make the more apoptosis of the back cells. So what happened? This apoptotic bleb, which is a PDCE2, which is from the mitochondria, you can see it is coming out on the surface. It is a releasing, and then it start increasing the anti-mitochondrial antibodies. So PBC patients have the AMA, and these antibodies, they make the apoptotic bleb much more, and which is due to this that there is a more of CD4, CD8, and TFH cell. There is an infiltration of pathogenic TFH, TH17, and there is ongoing back injury. However, you will ask that what were the regulatory T cells were doing. Regulatory T cells lose their efficiency. They don't inhibit this production of pathogenic TFH or the CD8 T cell, and there is an immune tolerance breakdown here. So susceptible and injured back start giving the apoptotic bleb. So however, in AMA negative patients, the clinicians are also taking care of anti-nuclear antibodies, which are SP100 and anti-GP210, both of which are associated with a more aggressive disease course and the poor prognosis. When there is a apoptotic bleb, which is coming on the surface, now there is a balance because there is an injury in the PBC, but now the balance is that whether all the backs will go to the apoptosis or there is a apoptosis or necrosis balance. So what happens in the cholangiocyte, there is already expression of TNF alpha. So, and TNF alpha is too much in the portal tract. So TNF alpha is a pro-inflammatory mediator. So all patients depend upon the whether there is a more of apoptosis or the healthy backs are still living and they are giving the proliferation. So there is a proliferation of residual cholangiocyte determines the evolution of toward duct loss and fibrosis. In advanced biliary diseases, cholangiocyte lose the ability to proliferate. There is this PDCE2, which is a moiety of the mitochondria. There is a big problem. This PDCE2 has the lipoil moiety, and this lipoil moiety is too much conserved in the bacteria, amoeba, fly, frog, mouse, and all human cells. This is a conserved moiety. Lipoil domain has in the PDC2 essential for the T cell epitope recognition. So when this is conserved, so you can't say that patient was genetically susceptible. That's the reason it got when I was reading the papers, even the papers have shown the PBC patient have the cause for the UTIs. They have the more of UTIs due to the bacteria inflammation, and because bacteria has the same lipoil domain, so they start inviting the T cells. Even the patients have the nail polish allergies because this nail polish also induce the PBC. So they have told that because of this lipoil domain, which is conserved, you can't say that the PBC is only with the genetic determinant. It is with the many things, bacteria and even the virus. PDCE2 homology with the virus make more. So virus incorporates in the PDCE2 and build from the cell surface, and it start attracting the local immune response to the viral proteins and along with the human PDCE2 at the lymph node and even in the biliary epithelium. So biliary injury does not stop here only with the immune cells. There is a disturbance in the bicarbonate umbrella when there is a PBC. How? This is a normal situation where is a ductal lumen in the apical basis, cell is loaded with the HCO3, which has the alkaline kind of the environment, and with the help of NHE, sodium and hydrogen even receptor, it extrudes the hydrogen and it contains the HCO3. So it is an alkaline environment of the back. However, in the PBC, what happens? This alkaline environment is disturbed and there is a molecule AE2 receptor anion exchanger. There is a reduction in anion exchanger. It happens that it is extruding the HCO3 more and it is taking the chloride ions more. So this, how it start disrupting, because this AE2, which is an anion exchanger in the PBC patient, in the normal, what happened, this is taking out the HCO3 and it is making the bile acids environment very much alkaline, and there is a normal biliary epithelial cell and normal biliary umbrella. However, in PBC, due to this modified PDC-E2, this HCO3 compartment is increased, and then the bile acids are here becoming, now when it is extruding, less of the HCO3 is excluding, it is more, and the H ion makes the bile acid acidic, and in the apoptotic bleb also it is making the more of the acidic. Now this senescent apoptotic Beck, what it accumulate, it secrete more again of IL-1, 6 and all the chemokine ligands, which leads to the manifestation of the progressive cholestasis and the fibrosis. What is the importance of transporters? Everybody knows that hepatocyte, in the hepatocyte, cholesterol is making the bile acids. Now, this bile acid is going through the canaliculi to the outside, to the ileum, and it is accepting by the enterocyte through the ASBT receptor. And now this bile acid is conjugated with FXR, RXR, PXR, PPAR alpha, everyone, and giving the production of FGF, which is a fibroblast growth factor 15, and maintaining the balance of bile acid pool in the enterohepatic circulation. However, whenever there is a disturbance in the nuclear receptor, FXR, RXR, PXR, then there is a disturbance of FGF15, and there is a disturbance of CYP7A1, which is the controlling enzyme which makes the cholesterol from cholesterol to the bile acids. So bile acids are now then disturbed in the PBC. How they are disturbed? Because in generally PPAR and PXR, they make the co-receptor, they heterodimerize, and then they make the either co-repressor complex or co-activator complex. So now there is again balance between the nuclear receptors, how they will heterodimerize with which molecule, whether PPAR will FXR or TGR or RXR. So how they make the co-activator complex and then start creating the genes mutations or the gene expression and regulate the hepatocyte bile acid synthesis, regulate absorption and secretion of bile acids, and induce the much more bile acid pool. Another TGR5, which is the membrane receptor, it is not the nuclear receptor. When it combined with the FXR and activate, then these receptors activate the bile acid pool and regulates the inflammation. In 2025, they have shown that through TGR5, this process make the M1 to M2 no pro-inflammation, even the anti-inflammatory, and TGR5 promotes the secretion of chloride and bicarbonate, protecting the hepatocyte from the toxicity of the bile acids. So deletion of TGR5, if there is TGR5 is not there in the patients or even in the several mice models they have shown, towards the pro-inflammatory NK T cells which produce the interferon gamma and again disturb the balance and immune injuries there. Another one paper was showing that there is that goblet cells in the intestine, they start secreting the trefoils. They generally secrete the trefoils. Trefoils are the mucin-associated proteins involved in the maintenance of epithelial lining. But in the PBC, generally what they do, they stop apoptosis. See here. But in PBC, trefoils are not there. Trefoils, especially during in the small bile ducts, there is no expression of trefoils. That's the reason small bile ducts are more injurious than the large bile ducts. Intestinal trefoil factor production in response to damage is absolutely absent, makes such cell more vulnerable than those in the large bile ducts. So what are the therapeutic options? So as I already told you that this is more with the enterohepatic circulation. It means in the enterohepatic circulations, bile acid pool, which is 95% is going back to the portal blood and going back to the liver, but the 5% of the bile acid which left behind in the intestine, in the ileum, they are manipulated by the existent gut microbiota. So it's a reverse. Bile acids are manipulated by microbiota, and microbiota is influenced by the bile acids. So dysbiosis of gut microbiota, imbalance of bile acids, and immune-mediated bile duct injury constitute the triad of pathophysiology in the PBC. So there are several clinical trials going on which have the use of probiotics, prebiotics, and symbiotics, and they have shown that probiotic lactobacillus LGG enhance the bile acid secretion and reduce the hepatic bile acid synthesis, increase the FXR FGF15 signaling, which balance that the how much cholesterol will make the bile acid and bile acid pool will be maintained. There is another molecule LP2 lactobacillus plantrum, which is also in the clinical trial. However, the FMT will be the best, which people have not yet tried, and bacteriophage therapy in the PBC was the recommended in the through the mice models. There is, if there we want to protect the biliary bicarbonate umbrella, the clinicians are using the glucocorticoid receptors, and which promotes the PXR mediated detoxification of bile acids. There is a new molecule budesonide, which is the again receptor agonist. It increases the PXR mediated detoxification of bile acids again. And there is a vitamin D receptor also, which constitute the and constitutive androsten receptor. Activation of this receptor is associated with anti-inflammatory, anti-cholestatic, and anti-fibrotic effects. People have seen that when the VDR is combined with the RXR, it is activating the antimicrobial peptide gene expression, which reduces the hepatic CYP7A1 activity also. This was in the Nature Review Gastroenterology. They have given that vitamin D receptor therapy would be the best, which agonism of the vitamin D receptor. PPR agonisms are already in the clinic, which reduce the bile acid synthesis via suppression of CYP and deconjugate the toxic hydrophobic bile acid, facilitate their urinary and the fecal excretion. There is a few drugs of the fibrate drugs. I am not going to do this. This is the chart where people have used certainly the approved treatments are UDCA and OCA, but potential treatment which are in the clinical phase 2 and phase 3, they are PPAR alpha, PPAR delta agonist, all agonist, and ASBT inhibitors are also, they have proposed it. Now, in the last, I want to say, because I wanted to keep this first also, but PBC is in the women. Why there is a gender imbalance? Because cholangiocytes have the real expression of estrogen receptors. So through the estrogen receptor, cholestasis is seen even in the pregnant women and during the pregnancy also. There is a sex chromosomal changes. I was very, very astonished to see that in PBC, there is a X monosomy is more. The people have found that significantly higher frequency of peripheral blood cell involve a single parentally inherited X chromosome. So X monosomy is there in PBC. There is an endosymbiont theory of mitochondria. Probably everybody, especially basic scientists, knows that mitochondria evolve through the endosymbiont theory, that there was the archaea and there was the bacteria when they came together, and there was an evolution of mitochondria, and mitochondria contains the mother DNA, and due to this, it's a more of the PBC is in the women. There are numerous genes like the FOXP3, which is a specially T regulatory component, are coded on the X chromosome, and there are several X-linked immunodeficiencies. Thank you so much for your kind attention.
Very nice, Dr. Nirupma. Any questions and comment will you take? Any questions or comments? Looks like they are not.
Good morning, everybody. I'll be speaking about the immunobiology of a sister disease that Dr. Nirupa just discussed, the primary sclerosing cholangitis. And this is another biliary disorder, but there is a slight difference. It is a chronic inflammatory progressive cholestatic disease. But the difference from the biliary cholangitis is that in sclerosing cholangitis, both the intra and the extrahepatic bile ducts are affected, and both these diseases can eventually lead to cirrhosis. And there is a very classical bead-like formation in the PSC, which all of you would be already aware of. These slides you must have seen earlier when Dr. Dinesh was presenting, and as Dr. Nirupma said, that there's a bicarbonate umbrella which is maintained in the healthy state, but when there is a diseased condition, you have the immune infiltration, and there is a loss of this bicarbonate umbrella, because of which there is a leakage of the bile into the surrounding tissue, which damages the bile, which damages the cholangiocytes also, and this leads to the activation of the local myofibroblasts, leading to fibrosis. This eventually advances to a very classical onion skin-like fibrosis that is seen in the pathology. Now, coming to the immune components that are involved in PSC, the first, of course, are the macrophages and neutrophils. Talking about the neutrophils, the studies have shown that it actually is attracted by the bile salts themselves, and there is an animal model of MDR2 knockout, which is an equivalent of the MDR3 gene or the ABCB4 gene equivalent in the humans. And what has been seen that the neutrophil elastase activity is increased in PSC animals where these the bile salt pump was knocked out, but this elastase activity was reduced when an antibody against this particular molecule was given, and this brings the activity down to almost the normal levels. The alkaline phosphatase and ALT activity also came down with the application of this. The fibrosis was reduced in a model where, in the same MDR2 knockout model, where CCL2-24, which actually recruits neutrophils, was inhibited by the antibody CM101. So this makes neutrophils one of the targets of treatment for PSC. Ma'am, can you use the mic, please?
Why was the MDR2 model used here? What was what is the rationale behind that?
So the bile release is disturbed in this, and it is actually released without any control. When you knock out the ABCB4 or ABCB4 equivalent in animal models, the bile is spontaneously released, not on the bile side, but also on the parenchyma side.
So we could have
So that causes the
ligation model also.
Sorry, ma'am?
How will it be different if we use the bile duct ligation model here?
Ma'am, but this is a model which we really cannot pinpointing exactly what is happening, but then this is a model which actually mimics the pathology of the disease.
Okay.
Then we have the mononuclear phagocytes, the monocytes, macrophages, and dendritic cells. And in a very interesting study, the healthy and the PSC monocytes from the patients were taken and they were exposed to heat-killed bacteria. And what was found was that Candida albicans increased IL-1 beta. Candida albicans also increased IL-6. But this was not the case in the monocytes that were obtained from the PBC patients. And Enterococcus fecalis also had the same effect, but there was no response to E. coli. That means that these monocytes are responding differently to different pathogens that are present in circulation, in the enterohepatic circulation. Now, in under classical conditions, there is increased monocytes in the blood and relatively reduced monocytes in the liver. But there is a certain class of monocytes which is called this, which is carrying a CD16 protein. The CD16 protein is actually a cellular cytotoxicity inducer, and this was reduced in other diseases than PSC, and it was enhanced in PSC. Whereas in PSC, the number of monocytes was reduced in the blood, but was increased in the other diseases. Now, this CD16 and CD14 both are present on macrophages, and there was a shift in the advanced stage PSCs where the CD14 numbers were reduced, and in PSCs the CD16 numbers were increased, suggesting that there is the thrust from the monocytes to induced cellular toxicity in the adjacent cells. Another protein which receives the scavengers and manures on fungi is CD206, which was found to be increased in the PBC, but was much suppressed in the PSC. So, although there is a biliary aspect involved in this, in both the diseases, PBC and PSC, but their pathologies differ slightly and the immune responses also differ. Coming to a very interesting cell, the mast cells, which are perhaps one of the least studied cells in liver diseases. In control, we do see a presence of some mast cells as shown by the pink stain here for the tryptase enzyme produced by these. In PSC, it is much enhanced. The FXR also is activated because of FGF19 coming from the intestine. So this connects this brings to us to the same story that in PBC also and in PSC also there is a microbiota component that is involved. So, but this was not present much in biliary atresia or in NASH. In NASH, FXR was less. In biliary atresia, although some tryptase activity was observed, but the FXR activity was less. So in both these cases, the microbial component probably is less. But we already know that NASH is directly related to a dysbiosis in the gut. And there are different mechanisms then which are actually leading to the pathology of each of these diseases. In a control experiment, they found that in PSC, FGF19 was increased in comparison to PSC, which had a comorbidity of IBD also. But SHP, another CYP7A1, the cholesterol producing enzyme, was increased in PSC, but was lesser than those that had IBD. So there are 2 or 3 different kind of molecular signaling that are going on with PSC with or without IBD. When cromolyn sodium was applied in another animal model of MDR knockout model, there was a very clear suppression of histamines, c-KIT, kinase, and tryptase, and the collagen level was also low in the tissue, as well as was the SMA and the fibronectin levels. So this suggests again that the mast cells also need to be looked at and targeted for treatment of this disease. The innate lymphoid cells are enriched in the mucosal space, and they are marked by the CD57 and CD56 DIM proteins on the surface. CD57 proteins were low in PSC, suggesting that these cells were less, although the number of these cells correlated directly with albumin and inversely with bilirubin and alkaline phosphatase. Another protein which induces NK cells, the NKp46, was found to be reduced in the PSCs with IBD, but was enhanced in PSCs where IBD was not present. Coming to the unconventional T cells, the mate cells, the NK T cells and the gamma delta T cells, it was found that the mate cells were reduced in the biliary diseases, PSC, as well as IBD and PBC. And when E. coli stimulation was given, there was a little difference that was seen in the expression of TNF alpha and IFN gamma in these cells. Although when mate cells, they were stimulated with E. coli and an anti-mate cell protein, R1 protein was inhibited with an antibody, there was an increase in the expression of TN alpha, and which was further more increased which was seen to be more increased in IFN gamma, suggesting that the mate cells also have a certain proportion of contribution towards inflammation. Coming to NKT cells, in a model where oxazolone was given, which is an NKT cell agonist, there was an increased immune cell infiltration, and the amount of cholangitis was also increased. Alpha SMA was also increased, and CD1D, which is a protein present on NKT, was when suppressed. Then it was seen that ALT was reduced and the grade of inflammation was also reduced. Now, in a very interesting study done very recently, multiple patient bile samples were taken and were given to naive NKT cells. Naive means unexposed NKT cells. This is the cell line number, and their expression of IL-2 was assessed. It was found that all these PSC patients did not induce NKT cells equally, which means that there is a certain constitution of bile that is required for activation of immune response. When again the anti-CD1 antibody was given to 3 samples which showed the maximum reaction, it was found that there was a significant suppression of IL-2 expression, except for the bile sample from B9. Of course, they did not do any metabolome analysis to determine what is the molecular composition of or characteristic of this bile. Now, there are multiple studies. One study shows that there is a difference in the amount of the bile salts that is present and the kind of the salt that is present. So, in PSCs, generally glycocolate is seen increased, glycochino is increased, hyocolate is also increased, and glycoursodeoxycolate is also found to be increased in comparison to PBC or the controls. Now, in a very elaborate study published in Nature in 2019, about 29 different bile salts were tested on naive CD4 T cells to see what kind of effect these bile salts have on these cells. And their differentiation into either the pro-inflammatory Th17 or a regulatory Tregs was observed. And what was found was that there was a non-uniform pattern of Th17, whereas Tregs were pretty much the same in response to all the bile salts, but Th17 was most inhibited by the 3-oxolithocholic acid. Another isomer of lithocholic acid, the isoallolithocholic acid, it on the other hand enhanced Tregs. So, which means that besides ursodeoxycholic acid, which is also used, we can have other combinations of modified bile salts which can be used for treatment. Coming to the gamma delta T cells, again in animal models, it was found that the serum IL-17A was increased in the animals of the PSC model, and when these cells were suppressed with the T cell receptor of the gamma delta T cells giving their antibody, the alpha SMA, collagen, and TNF alpha was again suppressed, which again makes these gamma delta C cells a separate entity which can be targeted to regulate or suppress the inflammation in PSC. With the antibody, the fibrosis scores were low, and so was the IL-17 expression. Coming to the conventional T cells, a recent spatial transcriptomic analysis. When I say spatial transcriptomic analysis, it is SPATIAL, which means we take the tissue, we take a section, and we put it on a slide, and we do a RNA-seq on the slide itself. So we capture the RNA expression of different cells present on the slide at the same time. And by this, we can identify different RNA expressions according to the cell type that is present on the slide. And this is a direct approach to assessing what combinations of gene expression is occurring. So in the spatial transcriptomics, it was found that the T cells cannot be classified into a single type, but they actually form 5 different clusters based on their gene expressions. The red means the increased gene expression and blue means the suppressed. And what they found was that when their fraction was tested, was checked in the liver, it was found that 3 fractions were increased in the fibrosis, whereas the other 2 fractions were not increased. Now here comes the role of DCs, which I did not discuss earlier. DCs express a certain protein called CD39, which is an ectonucleoside. It is expressed on the cell membrane and works on outside of the cell, where it actually breaks the ATP and ADP down into AMP. Now, ATP itself is immunogenic and induces inflammation. So when a CD39 cell line was used, it was found that there is an increase in ATP and there was a T cell activation and Th17 differentiation. So hepatic collagen was increased in this. Ductular reaction was increased, serum ALT and TGF beta were also increased. In PSC, although CD39 IHC, when scoring was done, it was found to be lower than in PBC, which means that DCs are more active in PBC than they are in PSC. Coming to a very interesting correlation between the genetics of PSC and the immune inflammation, there is a protein called BAC2. The full form is basically the 2 domains, BAB and CTC domains, heterodimer 2. Now, this actually induces Th17 phenotype. So when a certain mutation is present, there is an increased expression of microRNA 4464. Now, this microRNA actually targets the BAC2 gene. BAC2 gene shifts the balance between Tregs and Th17s in favor of Th17s and make it a pro-inflammatory setup. Whereas when this SNP is present, this miRNA expression is more, which chews the reduces the BAC2 RNA expression, which actually shifts the balance back to the Treg types, and Th17 cells, they go down. So suggesting that these patients would be probably having a better will not have a worse outcome with PSC than those patients who do not carry this SNP. So this probably can be checked in these patients before the start of the treatment. Coming from the Th17, we come to the neutrophil loop, where IL-17 and TNF, they mediate neutrophil rolling and endothelial transmigration through upregulation of E-selectin and neutrophil chemokines. These neutrophils then release nets, and they capture pathogens or pathogenic material. And these traps, they contain alarmins and histones that skew T cell differentiation towards T17 phenotype back again. Now, this Th17 produce more IL-17 and further induce the recruitment of neutrophils. So this is a cyclic loop which actually continues in PSC, which advances the disease. Coming to the B cells and the autoantibody production, a comparison between NASH and PSC was made, and it was found that in most of the inflammatory signals, cells are expressing heavily in PSC except for Th1 type. In NASH also they were reduced, and they were much reduced early on according to the grade in the PSC. Now, hepatic B cells in, again, the MDR2, which is a very classical model for cholangitis. At 25 weeks, it was found that the IgG was increased. Autoantibodies for ANA were increased at 25 weeks, and the B cell activating factor was also increased. When an antibody against the B cell activator factor was used, the IgG was reduced, and so was the ANA. Surprisingly, in this model, the fibrosis did not reduce, probably because of the persistent activation of myofibroblast. Now, this brings us to talking about IBD and all. This brings us to the gut-liver axis. And if we see the top panel, we see that we do not have much of PSC in India. It is more of a Western or a Caucasian kind of disease, with some being present in China also. But when we look at the PSC with IBD, we certainly find India also shining up in the map. And there is a very high number of PSCs with IBDs ranging from 60 to 90%. In India, it is about 30 to 40% PSCs which have IBD. So IBD, we know that is related, like NASH, to the dysbiosis of the gut. And in IBD versus PSC IBD, when samples were taken, biopsy samples were taken, and their analysis was done through single cell analysis, it was found that two separate inflammatory clusters were observed in these cell types, and they related to the B cell activation, phagocytosis in one type, and mitochondrial and respiratory electron transport chain in the other type, suggesting that there is a difference in the PSC with IBD and PSC without IBD, how it manifests itself. And there was increased dysplasia in PSC, but not in IBD. Also, there was a heavy response in the response to LPS. Again, linking it back to the activation of the cells because of microbial proteins. When pattern recognition receptors were checked, it was found that in comparison to ALD, PSC also showed a similar expression of protein expression of the TLRs, except that TLR4 was far more enhanced in PSC as compared to ALD. And the same thing was observed in the RNA expression of these. But when there was relative mRNA expression of these receptors was checked on the biliary epithelial cells, it was found that TLR4, IL8, and TNF alpha were far more increased towards the later stage of the disease rather than the early stage of the disease, where they were very similar to the control types, which indicates that the disease is progressive and the patients are likely to come only when the disease has reached a severe stage. In a global microbiota analysis, it was found that the microbial diversity is reduced in PSC, whereas in PSC with ulcerative colitis, it was the least. It was increased with ulcerative colitis alone. And 4 bacteria, Fusobacterium, Enterococcus, Lactobacillus, and Streptococcus, were found to be increased in comparison to the controls or Crohn's disease in these patients. Again, this group analyzed PSC of different grades with IBD and without IBD, where they found that there is no difference. But then when the combined panel was taken, where the PSC whole was taken, it was found to be reduced in comparison to the bacterial diversity was found to be reduced in comparison to the control, which suggests that there is dysbiosis, but of a particular type, where most of the bacteria have gone down in their numbers. Now, they identified Veillonella akkermansia, Clostridium, Citrobacter, Oscillospira, and one species from Ruminococcaceae. Now, why these bacteria are important? Because these are the bacteria which are actually involved in suppressing the tight junction proteins. And the problem with the biliary diseases is that particularly PSC, that the tight junctions are lost or reduced between the cholangiocytes, which leads the bile, and there is a bicarbonate umbrella reduction, which leads to the excess of tight junctions to these bacteria which have reached the bile duct. Now they actually cleave the tight junctions, because of which the bile is able to escape out. So if there is a biliary tube, then a bile duct, then if the cholangiocytes are loosened, then the bile is likely to come out to all the spaces around the tube. And that is what actually gives rise to fibrosis all around it and leading to that onion skin-like pathogenesis. They tried to develop a score from all these 7 bacteria, and they found that AUC to be about 0.8 or 80% accurate, but of which Villanella alone was contributing to about 65%. When compared to ulcerative colitis, the results were almost the similar. Now, in MDR2, germ-free animals were used to see if what happens if we completely remove the bacteria from the animal and then induce cholangitis. So what was interesting was that this actually did not improve cholangitis, it actually worsened, and there was increased fibrosis in absence of the bacteria, which goes on to show that certain bacteria are involved which actually protect the exacerbation of the disease. What was found, on the other hand, was that as the disease progresses, in germ-free animals, there was senescence in the cholangiocytes. So this means that these microbes are inducing some kind of trigger which actually stops the cells from going into a senescent state. What this trigger is could not be identified in this paper and probably needs to be understood. So summarizing it, what we see is that there is a microbial trigger which goes into the liver via the portal circulation, and there is activation. Th17, TNF alpha, IL-2 and other immune cells are activated individually, and all of them have to be countered. One drug may probably be not a solution for this disease. This leads to reactive cholangiocytes, and they can either go into a senescent state or can recover into a normal cholangiocyte, but depending on the microbial composition that is present there. I would like to end this with the quick linkage that Dr. Nirupma also suggested, that age and sex also has a role to play, because most of these patients come between 30 and 50, 55 years of age. So that means this disease is progressing very slowly. There could be disease subtypes depending on the genetic makeup of the individual, the lifestyle that individual is having, how rapidly the dysbiosis occurs or not, what is the association with IBD, and there are certain malignancy risks also. There are certain lacunae, of course, at the end of the liver, how the different cells are interacting with each other, which parts of bile duct are affected much more readily, why only beaded structure, why not a continuous fibrotic structure. Then what is the exact role of gut? Does SIBO have the small intestine dysbiosis has any role, or it's only the colonic? The immune cells and their exact migration pattern and the roles, and what are the other signals which could be coming from bone marrow, as we have seen in ACLF. So with these lacunae, what I think the way forward is that the leverage should be given to biobanks for preclinical detection, where we can do some rapid studies. Then spatial profiling, where spatial technologies to map immune microenvironment at single cell resolution can be undertaken to determine the therapeutic targets. Subtype stratification, mechanistic studies on organoids, and some promising targets are neutrophils, IL-17, Th17 axis, B cells, phagocytes, and of course the gut microbiota. And with that, I end my talk and wish you a happy new year.
Thank you. I think very nice, and I think little complicated, and I know these are challenges with these diseases, and we always clinicians struggle in that. Any comments or anything? Dr. Vijay?
Yeah. very elaborate. Rather, I will say the overdose of PSC and the immune system. I have two questions. One relates to your table where you show a study on a number of bile acid derivatives on Treg and Th17 cells, and you showed that one of the derivatives is having either is suppressive or proliferative action, an inhibitory action. So I would like to know that whether this study was done on a one concentration of all these derivatives, and if so, then what is the physiological level of these derivatives in the body?
Physiological level of these derivatives
Concentration. Yeah,
in sir?
In the body.
in the body. Because see, if you are studying in vitro, that is not a true reflection.
I am definitely not the best person to answer immunological questions, but the Tregs are the regulatory cells and have been known to suppress not only T cells but other cells also. So it is actually a balance between the intensity of the disease and the population of the cells that is available at the early stages. And as was seen that initially the levels of all these cytokines is low, but then it increases as the disease progresses.
No, I'm talking about the level of these bile acids, not their regulatory role on cells. What is the level of this physiological level? Because you may show in the lab that one is inhibitory or one is kind of
No,
stimulatory.
so that information is missing. That
Okay.
information is missing.
So second question, I think a quick one, that what is the trigger for mate cell, for differentiation of mate cells and proliferation?
It is the bile salts. So bile salts are kept in bile ducts for a reason. If they come out in open, they trigger the immune cells. And that is what happens when the tight junctions break. These bile salts escape. They trigger the local immune cells, which recruit the cells from circulation.
Right. Thank you. Thank you, Lakshota. And wishing everyone a happy new year.